Okay, hello everybody, and welcome to the webinar on hemoglobin and blood and dissociation curves. If you've been waiting, then hopefully you've gone into the Socrative room and taken part in the quiz, which should stimulate a bit of thought. Next webinar is capturing light, splitting water, light-dependent and light-independent reactions in photosynthesis, so looking at the Kelvin cycle and how you capture carbon and what you do with it. You can register on the website. Any comments, just drop me an email at learn@alevelbiologytutor.com. If you found it useful, then please do recommend to your friends or share with people, more the merrier. And also, if you could take the time to fill out the survey that I've emailed out to the mailing list and is also on my website, then it will give me a chance to plan the activities for last minute revision, which I'm planning on doing for the weeks before your exams, starting in the first week before the first exam. And then you've got them, I think, on 3 consecutive weeks, on the Monday, the Tuesday, and then again on the Monday. Beautiful day, and thank you for spending the time revising something that many of you find it quite difficult. Any points that you want to raise throughout this, there's a Q&A box, so feel free. Okay, so let's make a start. So we're looking at the carriage of oxygen and the carriage of carbon dioxide. Oxygen diffuses in the lungs, joins with hemoglobin, and then diffuses out into the tissues, where most importantly it is turned into water, where it combines with hydrogen ions and electrons at the end of the electron transport chain. So quick reminders about blood. Blood contains cells. It contains erythrocytes, which are the red blood cells, leukocytes, which are the white blood cells, and cellular fragments called platelets. And it also contains a fluid called plasma, which is water and the products of digestion and hormones and plasma proteins. So the plasma dissolves a bit of oxygen, a little bit of carbon dioxide, salts, glucose from digestion, fatty acids, amino acids from digestion, hormones, and big plasma proteins, which lower the osmotic pressure of the blood, which is important for tissue fluid formation, which I think we've talked about before. Okay, so how do you carry oxygen? Well, we're going to talk about this in a lot of detail, but just going over some of the major points now. So 99% or 98.5% of oxygen is transported within the erythrocytes, and it's bound to hemoglobin. Only 1.5% is dissolved in the plasma. It is just dissolved in the water. So hemoglobin is a complex protein. It's a tetramer. It means it's got 4 subunits. Each subunit contains a hemoglobin chain and a globin chain, and a heme group. And the heme group contains a single ion. Now, the ion is the one that has the affinity and can hold a single oxygen molecule. Because there are 4 subunits, there are 4 heme groups, therefore there are 4 ions. Therefore, each hemoglobin can carry 4 oxygen molecules. So when we take hemoglobin, we add 4 oxygens to it, we form oxyhemoglobin. Now, concept of partial pressure is one that students do tend to struggle with. And why it's so important is that the ability of hemoglobin to take up and then release that oxygen depends on how much oxygen is surrounding the hemoglobin. So, and the way we measure the amount of oxygen that's surrounding it is as the partial pressure, or ppO2. And this is measured in pressure, and the units are kilopascals. So ppO2 is a measure of the concentration of oxygen. Now, partial pressure is the part of an overall pressure that's caused by one gas in a mixture of gases. So therefore, there are 2 ways that you can change partial pressure. Either you have more or less air, which changes the total pressure, or you have more or less percentage of oxygen in that air. So sea level, the pressure of air is around 100 kilopascals. Oxygen is around 20% of that 100 kilopascals. Therefore, the ppO2 is around 20 kilopascals. Now, in the lungs, the oxygen is about 13% in the alveoli. Therefore, it's about 12% kilopascal partial pressure in the alveoli, which is where the gas exchange occurs. Remember, oxygen is being used in the mitochondria and turned into water in oxidative phosphorylation. Therefore, the partial pressure falls in the tissues because of aerobic respiration. Now, the more aerobic respiration you have, because there is a greater demand for ATP to power, for instance, muscle contraction or any other process that requires ATP, then the lower will become the partial pressure of oxygen, because the more the oxygen is being used for respiration. Now, at altitude, you've got less air. You've still got a percentage of oxygen of 20%, but it's 20% of a smaller pressure. For instance, at the top of Everest, you've got a air pressure of 33 kilopascals. Therefore, the partial pressure in the lungs at the top of Everest is around 12% of 33. So it's a lot lower. Same percentage of air, same percentage of oxygen in the air, but a lot less air. Now, looking at some of the partial pressures that you get, so this is you in the room at the moment. This is the humidified air that's in your tracheae and your bronchi and your bronchioles, and this is the alveolar air. So the percentages here, in room air, we've got about 21% oxygen and 0.04% carbon dioxide. In the alveoli, you've got about 5% carbon dioxide because it's diffusing out of the blood plasma and out of the red blood cells and into the alveoli, whereas the oxygen is about 13 or 14%. Just to make this point again, you breathe in the inhaled air, so you've got about 21%, and well, 0.04 is about 0% carbon dioxide. In the trachea, it's picking up more water because there's water, dissolved water coming from the walls of the bronchi and also from the alveoli. In the alveoli, it's about 13% oxygen, about 5% carbon dioxide, because the oxygen is diffusing into the red blood cells and the carbon dioxide is diffusing out. Now, the exhaled air that you breathe out has got a higher concentration of oxygen because the exhaled air is being mixed with the fresh air that's in the dead space, which is the non-exchange parts of your bronchi and your trachea. So this is why the percentage of carbon dioxide that you exhale is 4%, but the percentage of carbon dioxide in the alveoli is 5%, and why the alveolar and the exhaled oxygen levels change. Okay. So this is the different partial pressures as you go through the circulatory system. So when we've got oxygen leaving from the right ventricle, well, not oxygen, sorry, red blood cells leaving from the right ventricle and then going up, you've got a partial pressure of around 40, and this is in millimeters of mercury, not kilopascals. It then goes in and you've got a gradient to diffuse from the alveolar air into the blood and for the carbon dioxide to diffuse out, because this is the gradient here. Comes down the pulmonary veins, not an exchange surface, goes out through the aorta, again, not exchange surfaces, down the systemic capillaries, and then at that point you've got a gradient of partial pressure in the systemic arteries going to the systemic capillaries of 100 to 40, so the oxygen will diffuse out of the blood and into the tissues. And then the carbon dioxide will diffuse the other way and back into the blood. Okay, so how do you carry oxygen? Let's have a look at this in some detail. So the hemoglobin picks up or associates with oxygen to form oxyhemoglobin when you have high partial pressures of oxygen in the lungs. So the partial pressure of oxygen in the lungs is around this much, so we'd have a saturation of the hemoglobin of about 95%. And it dissociates, the oxygen dissociates from the hemoglobin in tissues which have a low partial pressure of oxygen because they're respiring. Now, the degree to which the oxygen comes off the hemoglobin is affected by the concentration of oxygen that's around it. So that tissues that are respiring a little bit will have partial pressure of, say, 7 kilopascals, whereas tissues which are respiring a lot might have partial pressures of around 4 kilopascals. Therefore, more oxygen comes off and dissociates and goes into tissues which are respiring more. Now, this curve is referred to as a sigmoid curve because at low partial pressures, the hemoglobin doesn't take up oxygen very readily, but as the partial pressure increases, all of a sudden it takes up a lot of oxygen very rapidly for a very small change in partial pressures. Now, this is a sigmoid curve rather than a straight line, which you'd expect. Now, this is because the heme groups are in the center of the oxygen, which makes it difficult for the oxygen to bind with them. So they've got at low partial pressures of oxygen, you've got a low percentage saturation of the hemoglobin with oxygen. Now, as the partial pressure increases, the diffusion gradient into the hemoglobin increases. Eventually, an oxygen will bind to one of the heme groups. Now, this is the crucial part to understanding why it's a sigmoid curve, is that the shape of the hemoglobin is dynamic. The shape of the hemoglobin is affected by how much oxygen is bound to it, and this is known as cooperative binding. So that when one oxygen binds to the hemoglobin, the whole hemoglobin molecule changes shape, which makes it easier for more oxygens to bind with the other heme groups. Now, here is hemoglobin. We've got a heme group with iron in it, and there are obviously 4. There are 2 alpha chains, which are polypeptides, and then 2 beta chains, and making up your tetrameric protein, which has got a quaternary structure because it's got more than one polypeptide chain. Here's another pretty picture. Again, heme groups here, 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 4 heme groups, and here's another pretty picture. Now, it is crucially important to remember that when you get one oxygen binding, the whole hemoglobin molecule changes shape slightly, which makes it much easier for the next one to bind, which makes it easier for the next one to bind. And finally, it's quite hard for the last one to bind because there isn't much of a concentration gradient at that point going into the hemoglobin molecule. Interestingly, if you look at heme and chlorophyll, they're almost exactly the same. Thought you'd like to know that. Now, understanding the partial pressure saturation graph, students, I think, fail to accurately realize what the graph is showing. So what they do is they get different partial pressures of oxygen and they bubble it through blood. So you might get a partial pressure of 1, you bubble it through blood, and you measure the percentage saturation, which is how much of all of the oxygen that the hemoglobin could be carrying, how much is it carrying? And then you draw a cross. And then you increase the partial pressure, you bubble it through the blood, and you draw a cross, and so on. So you repeatedly bubble different concentrations of oxygen through the blood, and you see how much oxygen the hemoglobin is carrying compared to how much it could be carrying. And that gives us the saturation curve. And the range in us goes from 0 in really active respiring tissues, so you're sprinting for a bus because you're using up lots of oxygen to power aerobic respiration, to 12 kilopascals, which is the partial pressure in your lungs at sea level. So when you bubble 1 kilopascal partial pressure blood through the oxygen through the blood, you get this point of saturation. It's very unsaturated. When you bubble 12 kilopascal, you get highly saturated blood, which is carrying as much oxygen as it can. So just to go over these points again, because I know it's something that's a bit of a challenge sometimes. Low partial pressures of oxygen, the hemoglobin doesn't take up oxygen very readily. And that's because when you've got, say, a partial pressure down here of about 3 kilopascals of oxygen, then 1, 2, 3 of the possible binding sites that are shown here. So we've got 3 oxygen molecules, therefore we've got 12 binding sites. If 3 of those 12 are bound to oxygen, then the saturation will be 25%. Now, as a result of those oxygens binding, the whole shape of hemoglobin changes, which makes it easier for the next oxygens to bind. So now, when we got 8 kilopascal oxygen, then more of the oxygens are going to bind to more of the binding sites. So now we take up 7 out of the 12, which is 58%. So we've increased the saturation here to 58%. If we increase to high partial pressures, very high partial pressures of 11 kilopascals, then more of the oxygen is going to diffuse in. And now 10 out of the possible 12 binding sites are taken. So now we've got a saturation of 83%. Now, it's difficult for all of the hemoglobin molecules to become 100% saturated because there isn't much of a diffusion gradient for the oxygen to bind in to associate with the final 4th hemoglobin, 4th heme group. So we see saturations of around about 96, 98% when you've got 12 kilopascal partial pressure that you'd have in the lungs. So this is the final one, high oxygen partial pressures, we'd have it almost all bound. Right, talked about that, right. Now, remember in the lungs, the oxygen associates with the hemoglobin because it loads up, so it's almost 100% saturation, whereas in the respiring tissues, the oxygen dissociates and is released. We're now going to have a chat about carbon dioxide transport. Understanding carbon dioxide transport is absolutely crucial in understanding the Bohr effect, which is the decrease in affinity of hemoglobin for oxygen as you change the pH. So we're going to look at the effect of high partial pressure of carbon dioxide, and we'll look at the transport of carbon dioxide. So carbon dioxide is produced in the respiring tissues, and it's produced by the decarboxylation of carbohydrates and fats in the link reaction and in the Krebs cycle, which occurs in the matrix of the mitochondria. 5% of the carbon dioxide is dissolved in the plasma, 10% combines with the hemoglobin to form carb-aminohemoglobin, but 85% is transported in the form of hydrogen carbonate ions, and we'll talk about how they're produced. Now, carbon dioxide is made in the matrix of the mitochondrion. It diffuses from the cytoplasm into the plasma because there is a concentration gradient, because it's being made in the mitochondria. So it diffuses into the cytoplasm, then from the cytoplasm into the plasma. That carbon dioxide then diffuses into the cytoplasm of the red blood cells. The carbon dioxide then reacts with water to form carbonic acid, and that's catalyzed by an enzyme called carbonic anhydrase. Now, carbonic acid dissociates to give you hydrogen ions and hydrogen carbonate ions. The hydrogen ions lower the pH, and these hydrogen ions then join with oxyhemoglobin to form hemoglonic acid. When you get to the alveoli, the whole process is reversed because you've got a low partial pressure of carbon dioxide, so that the carbon dioxide then diffuses out of the plasma into the alveoli, and the whole process runs in reverse, because remember, enzymes can work in both ways. So the hydrogen carbonate ions that you form, because you've made a load of them, there's lots of them. So there's a concentration gradient, so they diffuse out. Now, as you've got a lot of negative charges diffusing out, what you do is you would create a electrical gradient, so the chloride ions diffuse in to balance the loss of the negative charges. The hydrogen ions join with the oxyhemoglobin to form hemoglonic acid, and the chloride ions diffuse back in. Right, I've got a better on this. Right, respiration. Carbon dioxide diffuses into the red blood cell. Carbonic anhydrase turns it into carbonic acid. Carbonic acid dissociates. The hydrogen ions join with oxyhemoglobin to form hemoglonic acid, and the hydrogen carbonate ions diffuse out down their concentration gradient from where they're being made, which is in the red blood cell, to where there are fewer of them, which is in the plasma. The chloride ions diffuse in to balance that loss of negative charges, and then the hydrogen ions join with oxyhemoglobin to form hemoglonic acid and release the oxygen. The oxygen then diffuses into the tissues and diffuses into the mitochondria, to the inner mitochondrial membrane, where it joins with hydrogen ions and electrons and forms water, which is because it's the final electron acceptor in the electron transport chain in the mitochondria. Because it's being consumed, because the oxygen is being consumed, this maintains the concentration gradient going into the tissues, because it's being used for respiration. And because the carbon dioxide is being produced from respiration, this maintains the concentration gradient going in, plus it's being turned into hydrogen carbonate ions, which then diffuse out. And this is how you carry 85% of the carbon dioxide is as hydrogen carbonate ions. Now, the hydrogen ions would lower the pH, and these would affect the enzyme activity. So hemoglobin is a buffer. It absorbs the hydrogen ions, forming hemoglonic acid, HHB. The hydrogen ions change the affinity of hemoglobin for oxygen. So they change the shape of the hemoglobin molecule because they bind to the amine group, the N-terminus group, and to the, I think it's the histidine amino acids in the beta globin, and that changes the shape of the hemoglobin so that oxygen comes off. So it's lowering the affinity of hemoglobin for oxygen. And that oxygen comes off, goes into the tissue fluid, then diffuses into the cells. So the reason for these hydrogen ions is to cause the hemoglobin to be less saturated with oxygen than they would be normally. And this is called the Bohr shift. Now, this is an extremely important adaptation because it means that tissues that produce lots of carbon dioxide, which are the tissues that are respiring a lot, will lower the pH of the red blood cells that pass through them. Therefore, they will decrease the affinity of the hemoglobin for oxygen. Therefore, more oxygen will come off the hemoglobin and go into the tissues that are doing the most respiration, which is a terrific adaptation. So the more the cells respire, the more carbon dioxide is produced, the more hydrogen ions push more oxygen off and into the respiring tissue. Therefore, cells that require the most oxygen receive it because they're the ones doing most respiration and producing the most carbon dioxide, therefore lowering the pH. Isn't that amazing? So very often you see this called the shift to the right, and I've kept it as a shift to the right. What it actually is a shift downwards. So if we've got A, which has got a low partial pressure of carbon dioxide, B's got a medium, and C's got a high. So the more we increase the partial pressure of carbon dioxide, then obviously the more hydrogen ions you're going to produce, because the more carbonic acid is going to be produced because you've got a higher concentration of carbon dioxide. Then the more oxyhemoglobin is going to be turned into hemoglobinic acid. Therefore, the more oxygen will dissociate from the hemoglobin for the same partial pressure of oxygen. So the higher the carbon dioxide level, the more hydrogen ions will displace the oxygen, more oxygen will come off and will dissociate and go into the respiring tissues. As the blood enters the respiring tissue, the oxygen saturation of the hemoglobin is high. However, the partial pressure of the respiring tissue is low because it's using up the oxygen. The oxygen hemoglobin releases more oxygen to the tissues, blah, blah, blah. Final effect, more oxygen is available when more carbon dioxide is produced. Therefore, the more you exercise, the more you get supplied with oxygen because the more carbon dioxide you're producing. This is a sort of classic graph that you get in exam questions with three different dissociation curves of three different concentrations of carbon dioxide. So if you can see for a partial pressure of 4 kilopascals, then at high partial pressures of carbon dioxide would be having a percentage saturation of 20%. At medium levels, it's about 35%, and very low levels of carbon dioxide, it's about 80% saturated. So the difference is that when you increase the partial pressure of carbon dioxide and you have a high partial pressure of carbon dioxide due to actively respiring tissues, then the saturation of hemoglobin with oxygen will fall from 80% to 20%. So for the same partial pressure of oxygen, you'll be releasing 70% more oxygen into the tissue from that hemoglobin. So a couple of questions here. Explain the circumstances under which the partial pressure of oxygen may fall to 3 kilopascals in muscle tissue. So what they're looking for you to say here is to link the fall in partial pressure to the degree of activity by muscle. So an example that you could say here would be running or walking, running lots, any activity that would result in the consumption of lots of ATP. Therefore, you'd have to produce more ATP through aerobic respiration, which would consume the oxygen, which would cause the partial pressure of oxygen to fall. State which of three curves represent the hemoglobin with the lowest affinity. Well, that's C. If the pH of the blood became more acidic, the position of the curve for the mouse would change. Draw on the graph opposite the new position. Well, if it becomes more acidic, if that's the curve we have originally, then when it becomes more acidic, it's going to move again down further. And explain the benefits to the mouse of this change is that the tissues in the mouse that are respiring the most actively are going to be the tissues that receive the most oxygen, because the affinity of the hemoglobin for oxygen will fall when the blood becomes more acidic as a result of the dissociation of the carbonic acid. Okay, now moving on to fetal hemoglobin. Now, remember that fetuses can't breathe. So when you have a mum and you've got a baby, well, the way that the fetus is being supplied with blood is supplied with oxygen is from the lungs of the mother. So any oxygen that's received by the fetus has to join with the maternal hemoglobin and then has to be pumped to the placenta, which is an exchange surface with the fetus. Now, in order to get the oxygen to go onto the fetal hemoglobin has to have a greater affinity for oxygen than the maternal hemoglobin. And it does this by having some different proteins. So remember, in the placenta, the blood flow close together the maternal and the fetus blood flows close together, but they don't mix. It's not maternal blood flowing around the fetus, it's fetal blood, and there is an exchange surface. So here we've got the fetal capillaries, and they sit in a vat of maternal blood. These are known as sinuses or lacuna, I think. Lacuna. So there is maternal blood here, and oxygen is going to be diffusing from the maternal blood onto the fetal blood. And it only can do that because the fetal blood has a greater affinity for oxygen. So this is adult hemoglobin. We've got 2 alpha and 2 beta chains, and with fetal hemoglobin, we've got 2 alpha and 2 gamma chains. So it's a different, 2 different proteins, 2 different polypeptides within the protein. Now it says it's a shift to the left, and most of your books will say that, but the other way of looking at it is the shift upwards, because for the same partial pressure of oxygen, fetal hemoglobin is going to be more saturated with oxygen than adult hemoglobin, which means that if you had a partial pressure of, say, 7 kilopascals in the placenta, well, if the fetus had adult hemoglobin, then the blood that would be leaving the placenta would only be 60% saturated. But because the fetus has fetal hemoglobin, then with a greater affinity for oxygen than adult hemoglobin, then the blood leaving the placenta is around 80% saturated. So this means it's carrying a lot more oxygen than it would do otherwise if it had adult hemoglobin. Also, fetuses have a lot more hemoglobin in their blood than adults do. So it's got a greater affinity for oxygen. It picks up oxygen, and the mother's blood is releasing the oxygen. And in the placenta, the fetal hemoglobin binds to oxygen at a lower partial pressure than it would do if it had adult hemoglobin. So the fetal hemoglobin is more saturated as it leaves the placenta than it would be if it was adult hemoglobin. So it's shifted to the left. I prefer to look at it as a shift up because it's got an increased affinity for oxygen. I think I've said all of that. Myoglobin, some of you know about this, some of you don't. So myoglobin is what you get in muscles. Myoglobin is a monomer, not a tetramer. So myoglobin only contains 1 oxygen associated with the globin chain and the heme group. It's got a much higher affinity for oxygen, so it stays. The dissociation curve for myoglobin is like this. So it doesn't dissociate unless the oxygen partial pressure gets very, very low, like it would do in exercising muscles. Myoglobin doesn't go around in your blood, it stays in your muscles. It's a store of oxygen. And you give up that oxygen only when the partial pressure gets very low indeed. Another one that you tend to see is lugworm dissociation curves. This is in some syllabuses. Lugworms are annelids, they're worms, they have hemoglobin, but their hemoglobin only dissociates at very low partial pressures. This reflects the way of life of the lugworm. So this is your lugworm, and you see these casts that are produced in the sand where they build burrows, and when you walk down to the low tide mark on a sandy beach, you can see the exposed in the sort of last hour of the tide going out, or the last couple of hours. So these are adapted to living in habitats where there are low levels of oxygen, they've got low metabolic rate. When the tide goes out, they're trapped with water in the burrow. It's got a limited amount of oxygen, so it loads oxygen at very low partial pressures because it's very much to the left, because the curve is down here. and then it unloads the oxygen in the tissues. Exactly the same, but because it hasn't got much oxygen in the burrow and so on. Not a lot of people have got to cover that, but there we go. Llamas, you do need to know about for quite a lot of specifications. So this is a llama. This is the best picture I could find on Google. Now, llamas live at altitude. So they live in the Andes at high altitudes. Now, the partial pressure of oxygen at high altitude is lower than sea level because there is less air. It's still 20%, but the oxygen level is still 20%, sorry, but it's 20% of a lower number. So when you look at llama hemoglobin, it's got a dissociation curve which is to the left of animals which live at sea level. And the advantage to the llama is that if it's at altitude, so at sea level, the lowland animal is going to have 95, 98% saturated hemoglobin as it leaves its lungs. However, if the sea level animal went to altitude, then the saturation curve of its hemoglobin is significantly below the level of the llamas, because the llamas have got a greater affinity for oxygen in their hemoglobin, which means that at low partial pressures, which they experience at altitude, they can load more oxygen onto their hemoglobin than they would do if they had the hemoglobin of a lowland animal. Right. So the llamas possess hemoglobin which loads oxygen more readily with oxygen in the lungs than the lowland animals. The curve is to the left. Therefore, the hemoglobin of llamas is more saturated at altitude than would be the hemoglobin of the lowland animals if they were at altitude. And the advantage to the llama of this is that more oxygen can be supplied to the tissue of the llama because it's got this different hemoglobin with a greater affinity for oxygen. Okay, that's the end of today's hemoglobin dissociation curves webinar. Hope that helped. I hope that made sense. Any questions at all, please go ahead and ask them, and I'll try and answer them now. I can see a couple of questions have already come in, and I'll address them now. Next webinar is the one on photosynthesis, which you can register at alevelbiologytutor.com. And any comments, please email me at learn@alevelbiologytutor.com, and please fill out the survey. And the questions I'll address now is, why does oxygen travel at low partial pressures in animals where oxygenated and deoxygenated blood is mixed? Some frogs. You've got, so Hasith, I will address that in a minute. How does it have 12 binding sites? It doesn't. It has 4 binding sites, but I was just showing 3 molecules of hemoglobin that would have 12 binding sites in total, to get you an idea of the percentage saturation, because some students get slightly confused as to why it doesn't go up in 25%. So if you show 3 molecules, then they would have 12 binding sites. Why was hemoglobin called a buffer? It was called a buffer because it absorbs the hydrogen ions, therefore it means that the pH doesn't change. So a pH buffer, the pH buffer is a solution that maintains a constant pH, and the hemoglobin is acting as a buffer because it's absorbing those hydrogen ions and they're being turned into hemoglobinic acid. The next one said, will the discount code be the same for all of the webinars? And the answer is yes, it will be. I've emailed out a 20% discount code for the future webinars, and that will be applied to all of the future ones. Question regarding transport of carbon dioxide. When there is a high hydrogen ions, does it react with the oxyhemoglobin? Yes, it does. So when there's a high concentration of hydrogen ions, it reacts with the oxyhemoglobin and causes a change in shape of the hemoglobin. It turns it to its I think is it the R or the T state, but it's the state where it has a reduced affinity for oxygen, so the oxygen comes off. Down at the bottom. So doesn't this mean that the oxyhemoglobin acts as a buffer and not the hemoglobin? I suppose it does. Yes, that's a good point. Well done, Jay. What's meant by positive cooperativity? I'm not sure, and I don't think I used that, but what the concept that you've got to get with hemoglobin is the fact that as oxygen binds, it increases the ability of the next oxygen molecule to bind, which is why you don't have a straight line, you have a sigmoid curve. Are your revision webinars before the exam just for A2? Anybody can come along, but the exams are all now linear. So I'm going to focus on doing all of the topics, and the A2 topics tend to be the more difficult ones for many students. More questions, people? Any more questions? Happy to answer any of them. Would you mind telling me where you all are? Because I know some of you are coming from a long way away. So maybe type in the country you're in at the moment. Any more questions, people? London, that's not very far. I'm glad you're here and thank you for joining us. Good point. Okay, so somebody said, why does blood travel at a lower pressure in animals where oxygenated and deoxygenated blood is mixed? The point there, let's just try and, I think you're conflating two different things. So I'll just draw you through this. So with frogs, you've got an atrium, get it right. So I'll draw it just as sort of plan here. So you've got an atrium and another atrium and then a ventricle, and I'll do it coming out the bottom just to make it clear. And then this goes up and it splits, and then some of the blood goes round the lungs, doing this from memory, and then some of the blood goes round the rest of the frog. And then the blood goes back into the atria, and the blood that's gone around the frog goes back into the atria. So you've got some oxygenated blood going back around the lungs and some deoxygenated blood going back around the frog. Does that make sense? And so you said, why is the partial, because you haven't got two atria and two ventricles, you've got two atria and one ventricle. So the blood that's exiting here goes round the lungs and then go back in, and this is coming in at a bit of a lower pressure. Don't think I did very well with that one, but there we go. Shropshire. And you've said, how have athletes' hemoglobin adapted to travel to carry oxygen efficiently? They have exactly the same, athletes have exactly the same hemoglobin as we do. It's just that they may have more of it because they either have exercised at altitude, which has got a lower partial pressure, which causes you to produce a natural hormone, which increases the number of red blood cells that you have, or alternately, they inject erythropoietin, which is a drug, which causes you to increase your red blood cell production. So athletes have got the same hemoglobin as we do, they've just got more of it because they've got more red blood cells. Somebody said, going over the questions in the AQA book, and the question says, explain how DNA leads to different hemoglobin molecules having different affinities for oxygen. What would you need to answer this to get full marks? The concept that they're looking for there is that DNA codes for the sequence of amino acids. Therefore, when you have different globin chains, for instance, fetal hemoglobin has got the gamma globins and 2 alpha globins, where we've got 2 alphas and 2 betas. That's we as adults. So how you need to answer that is to say that DNA codes for the primary structure of the protein, and the primary structure of the protein determines the shape of the tertiary structure of the peptide, the polypeptide that gets produced as a result, and that would then affect the affinity of the hemoglobin to bind for the oxygen. So it's connecting a synoptic concept of DNA coding for amino acids in a protein to the folding of the protein, to the function of that protein in picking up oxygen. And if you change the amino acid sequence, you change the shape, and the way you change the amino acid sequence is changing the DNA sequence. Hope that answered your question. Can I look after this webinar in my own time, or will it be gone after it finishes? It should be on YouTube and also on my website, although I think the future ones I'm not going to do that with. But yeah, it should be available afterwards. What are good sources for practical questions or synoptic ones? My website and my blog are particularly excellent, I think. Other places you can look at are all of the old OCR unified biology, or not unified biology, unified concepts papers, which were from the specification before this one. I think the code is 2806. What is myohemoglobin? I think that's just made up, but I think myoglobin, myoglobin is a monomeric protein, so it's one heme group, one globin, and you find it in muscles, and it's a store of oxygen. Are all my past webinars on YouTube? Yes, they are. Alex says, can you also make future ones available? Well, I do want to charge for the future ones because I've been doing this for some time now, and it would be nice to get a little back from it. So I probably won't make the future ones available on YouTube because it would be nice if people paid a small amount of money to join in future. Sorry, Alex. I have emailed out a discount code, and I will give a discount code of at least 20% to anybody who wants to, anybody who's been to the existing ones. Will doing previous ISA questions be a good preparation for the practical side of the paper? Well, it's going to be very hard to prepare for the practical side. I think the major thing you've got to focus on is what is the independent variable? What's the dependent variable? How do you ensure validity or reliability? What's the difference between a control variable and a control? And then practice the math skills. When do you recommend to start doing past papers under time conditions? I don't know whether it's ever a particularly good idea. My biggest recommendation for all of this is understanding the concepts. The more you can understand the concepts, the easier the whole thing becomes. With hemoglobin, the concept is a small change in partial pressure of oxygen causes a large change in the affinity of hemoglobin for oxygen. So when the partial pressure changes because you exercise more or you consume more oxygen, then more oxygen is released. So when you start getting your head around that and head around cooperative binding, then that's the key to getting all of this, is to understand why things are happening and what benefit there is to the organism. Any more points, people? Do you think the grade boundaries will be low? Yes, definitely. Do you have any advice on how to prepare for the multiple choice side of the paper? So multiple choices in Edexcel and OCR. I've put four multiple choice quizzes on my website. I converted them with Google Forms, and they're from the sample papers for the AS sample paper and the 2A2 sample papers. I've got the practice paper set 1 and the practice paper set 2 multiple choice questions, which I'm going to load on probably after Easter, because I know lots of students are using them for, lots of teachers are using them for mocks, and I don't want to upset people. So I've got those ready to go after Easter, and also I've got video guides going through the OCR and Edexcel papers. So all of the sample papers are on my website to download. They're about an hour each, and then I've got the video guide to the OCR practice set 1 and practice set 2, all 3 papers. I will load those up probably in 2 weeks before the exam. What would you suggest the best way of revision for biology is? Understanding the concepts. Get a good textbook, watch some good YouTube videos, come to the webinars, and understand what's going on. There's a huge amount of facts to learn, but those facts are much easier to learn when you understand the concepts. Which textbooks would you say is best for OCR A? 2 textbooks I really like. I really like Campbell Biology, which is an American textbook. It's beautifully written and it's very good. It's best way to get it is on a Kindle, the Kindle download. It's about 40 quid. Well worth it, and you can treasure it for the rest of your life, or use it at university if you're off to do a course related to biology. The other one I particularly like is like this one, the Fullick book, which is the big thick one, OCR A, and Fullick. I think it's quite well done, so I'd recommend that one. Any more questions, people? Anything at all I can help you with? Nope. Well, I hope some of you will join me in about a month's time for the photosynthesis webinar, and I hope you send me your suggestions as to what I should do by filling out the form on the website. Will the practice questions be similar in all boards? They're using the same specifications set by Ofcol or QCA, so yes, they are very similar. Or they'll be testing similar skills. The OCR ones seem to be particularly unpleasant, it has to be said. But there we go. Thank you, Jay. That's very nice of you to say that. So would doing different specimen papers, practical questions be useful? Yes, certainly. I think the OCR ones are particularly good. So yes, I would thoroughly recommend doing other different specimen papers for 2017 rather than doing the old spec of what you're currently studying. Do I have more of those synoptic questions from the 90s? Yes, I've got loads of them. I'll put some more on the website. And I've also got some extremely good ones, Nuffield questions, which are about 20 years old. There's lots on the blog, and if I've got time, I'll load some more on next week. That's very nice of you to say that, Meg. Thank you very much. Have I done a webinar for tissue fluid yet? I don't think I have. It wouldn't last very long because tissue fluid is just squeezed out by the hydrostatic pressure at the arterial end and then returns in at the venous end because of the lower osmotic pressure because of the plasma proteins, which are too big to get through the epithelial layer. I'm pretty sure I've got, if you look at my YouTube channel, I think I did something on tissue fluid. So if you have a look at the YouTube channel, there should be a video on that. Have you done one for the kidneys yet? Yes, I have. It was very good. And I think I forgot to record it, which is a shame. I'll probably do another one though. Would you be able to do a session on biodiversity and population sustainability, please? Oh, no. I mean, it is mind-numbingly dull. I just really couldn't face doing an hour on that. So biodiversity possibly, but population sustainability, it's very, very boring. Yes, it is. It's environmental science as far as I'm concerned.